This is my model of polypeptide synthesis. I'm just going to be showing you a couple of things and then I'll be explaining it. So stage 1. RNA polymerase joins itself to the DNA at the promoter sequence region, breaking the hydrogen bonds that is reinforced through the nitrogenous bases. The outcomes, loosening of a part of DNA, double helix, so this is the double helix here, and this interaction is known as initiation in transcription, not translation. We'll get into that later. But it is important to note that RNA polymerase does not unwind the entire DNA molecule, only a section consisting of the gene that is required to code for a particular polypeptide chain like in here. The section is allowed to unwind to allow free ribonucleoside triphosphate molecules to perform complementary base pairing. So that's what's happening over here for a particular for a template strand of the DNA. So that's the one here. There is no thymine base in ribonucleoside just because they're replaced with uracil, so there's no T's. There's no T molecules and now U molecules, and this is because mRNA molecules consist of uracil, adenine, cytosine, and guanine, where DNA strands consist of thymine, adenine, cytosine, and guanine. After that, stage 3 of this is basically the RNA polymerase moves downstream of the DNA from the 3 to 5 displayed in the graph. So you can see the 3 here, and then you can see the 5 here. It's also again here, the 5. And as additional, and then the free nucleotides found during the DNA replication is called elongation because more RNA nucleotides are being paired with the nitrogenous bases on the format strand, right? And as the RNA polymerase moves downstream of the DNA, so that's what's happening here, it's moving downstream, you know what I'm saying? The enzyme rewinds the DNA, as you can see here. So if you look up here, see how it's rewinded as soon as it comes out. That's being rewinded to remake the double helix, and then it goes on. And the transcription stops only when RNA polymerase gets to a termination sequence. So that could be like UAG, for example, right? So then we move on to translation. Translation is the second main part of polypeptide synthesis. And step 1 is when the mRNA moves out of the cell nucleus and into the cell's cytoplasm through the nuclear membrane pore. Now, I've labeled the nuclear membrane pore right here. So as you can see, it comes out of that and then it goes here. And then a small ribosomal unit attaches itself, and then a large one attaches itself. You know what I mean? And there are tRNA molecules found in the cytoplasm that have an anticodon. One anticodon is comprised of one of 3 nucleotides. Every RNA nucleotide has a nitrogenous base, either A, U, C, or G. So as you can see, it's either A, U, C, or G. And that just goes on. And every one of these tRNA molecules can bind with a particular sort of amino acid that matches for its anticodon. And then step 5, as you can see here. Oh, by the way, I forgot to mention with step 4, every one of these tRNA molecules might, as you can see, the free ones here, and then they start binding up here again. And then with step 5, the mRNA codon specifies the tRNA carrying an amino acid matching the anticodon. So that's what's happening over here. And it binds with itself, and the ribosome checks the mRNA codon so that the tRNA molecules with the right anticodon bind with the right mRNA codon. So it always has to be the right one, otherwise it literally just does not work. And as a result of this, every mRNA codon indicates a tRNA molecule based on the correct codon, so it specifies with the amino acid. And because of this, the amino acid is a specific for the mRNA codon. So like I said, this has to be very specific. This has to match up perfectly, otherwise it does not work. And then we move on to stage 6, which is as the following mRNA codon specifies itself, the one more tRNA to bind with it The earlier the tRNA molecule will cut itself off from the mRNA and transfer its amino acid to the new tRNA molecule entered through the ribosome complex. The amino acids go through a build-up stance in response to a bond by means of a peptide. As this elongation process of building the amino acid chain goes on, the ribosome unit moves along the mRNA to keep reading the mRNA codons. The prolongation interaction of building the amino acid chain stops when the ribosome complex reads an mRNA stop codon. So at this stage, a release factor comes in and binds with the stop codon, and this makes the amino acid chain separate from the tRNA molecule, making a polypeptide chain. Okay, so basically what has happened here is that here it's based on the correct codon, and then this one will cut itself off. So as you can see, it's moving out again, and it's going back into the cytoplasm. And then what happens here is that the prolongation interaction of building the amino corrosive reads a stop codon. So that's what you can see here, the stop codon. And at this stage, a release factor will come in and it'll make the amino acid chain separate, making a polypeptide chain, which is all this stuff here, right? And basically, the ribosome complex isolates itself into small and large ribosomal subunits, and the mRNA splits into singular nucleotides. So this is all these here. And the polypeptide chain and kills up as the amino acid structures hydrogen holding with one another. And now this single polypeptide, so there's only one single polypeptide, is now a protein. So that's basically the gist of it. Yeah, sorry my animation didn't work. I don't know what happened with that. But I'm gonna leave what I said in a doc here, just in case you want to like read it. If anything, I feel like if you didn't hear anything wasn't clear. That's basically polypeptide synthesis. I'll also leave some of these photos on the classroom. And yeah, that's basically it. Thanks for watching.